Hi, good evening, everyone. It's a great pleasure to talk over here. I am Narayana Komravelli. I'm a research scientist at Pediatrics Infectious Disease at University of Texas Medical Branch at Galveston. And our department is focusing on respiratory viral infections and its inflammatory pathways and cell signaling pathways in lung cells, and in the areas of respiratory syncytial virus and human metapneumovirus. My research project focus on the host and micropathogen interaction. That means how lung cells respond to the virus when it's infected inside, and it's the mechanisms involved in the lung cells. So briefly, respiratory syncytial virus, it's also called as RSV, is a major cause of upper and lower respiratory infections and bronchiolitis in children and at the age of especially infants to young children age of 5 years. It has a tremendous burden. In fact, respiratory infections altogether consist of considered as a third leading cause of mortality around the world. So, RSV is one of the leading cause of lower respiratory infections in children. In US alone, about 200,000 patients are children patients are hospitalized, and about 1.5 million patients outpatient divisions are there, and about 800 to 1,000 deaths every year, and with economic burden of more than $500 million. And throughout the worldwide, there are about 35 million patients are outpatients are there, and about 3.5 million hospitalized patients below the age of 5, and about close to 200,000 deaths all over the world, mostly in the developing countries. And RSV infections are also major concern in elderly population, majorly aged above 65 to 70 years. So these RSV infections or lung infections majorly come in usually chronic lung diseases, some has, or chronic heart diseases, or immunocompromised patients where their immunity to fight general viruses are low. So even in United States, about 6,000 elderly people die just because of respiratory infections, and that is a huge economic burden. And apart from this, though there is intense studies going on respiratory syncytial virus research from several decades, there is still no effective vaccine or drug against this virus. So our laboratory focus on the pathology mechanisms, how RSV does infections and in human lungs. So though several research has been going on, we do not know exactly how the mechanism of RSV infection takes place. So our lab focus on two areas of RSV infection. One is the inflammatory pathway, and other is oxidative stress. So these two are majorly involved in the lung tissues. So my research project focuses on the elucidating the mechanism of oxidative stress in lung cells, and if whether there would be any potential drug targets that could outcome into in my research study. So let me introduce briefly what is oxidative stress. You would have several times heard about what is oxidative stress, this stress, that stress, but oxidative stress is generally caused because of increased production of some free radicals. Let me give briefly. Oxygen is essential for our life. Every cell needs oxygen. I mean, when we take oxygen, our cells, lung cells, any other cell utilize oxygen and produce energy in the form of ATP. When oxygen is utilized, free radicals called ROS, reactive oxygen species, which has a like superoxide, which has an extra electron in their outer orbit. These are generated as byproducts. When these byproducts are free radicals in limited controlled manner, they are very good, essential. They help in communicating cells from one cell to other cell, and how the internal cellular signaling it would help. But however, when they are produced in large amounts, they become toxic, and they damage cells components like proteins, carbohydrates, DNA. When these ROS, like some of the examples I'll give, called superoxide dismutase, or hydrogen peroxide, hydroxy radicals, nitric oxide radicals. So these are very, very active, energetic molecules. They are ready to react with anything which come in their contact. I mean, since they are reactive, they take some electrons from these macromolecules, or they donate their electron, and they become stable. So usually our cells have a very good protective mechanism called antioxidant mechanism. So they have achieved by through antioxidant enzymes called superoxide dismutase, which neutralizes superoxide radicals, and catalase. This neutralizes hydrogen peroxide radicals. So through a variety of mechanisms. So all this at genetic level, all these antioxidant enzymes are controlled by a redox signal transcription factor sensor called NRF2, which senses if a cell has more number of free radicals, there is a changes in redox potential of the cell. So it acts like a molecular switch. So this NRF2 goes inside the nucleus, and it synthesizes, induces it switches on, like any electric switch. It switches on the antioxidant gene synthesis. So when these antioxidant enzymes produced in the cell, and they neutralize all the free radicals, once the redox status is achieved, this NRF2 switches off these genes to maintain the cell's energy. So when there is an increased amount of production of free radicals, and there is no balance of these antioxidant enzymes, there would be a stress called oxidative stress would be generated. So these free radicals at molecular level bombard with whatever comes into contact. Like if protein come into contact, they inactivate protein, or they alter the function of the protein. When they react with DNA, they damage DNA, so that there would be altered synthesis of genes. Or if it bombard with lipids usually present in the cell membrane, so it changes the permeability of the membrane. So that's how these free radicals damages. So in case of RSV infection, when RSV enters into the lung cells, so this infection induces a lot of free radicals in the form of hydroxy radicals, superoxide radicals, nitric oxide radicals. So we investigated whether, when the cell has its own antioxidant enzymes, what they are doing, why they are not neutralizing. So when we looked at the levels of all these antioxidant enzymes, we noticed that antioxidant enzymes are significantly reduced, and so that free radicals are not neutralized, so that there is a stress. So then we put a question: how come these antioxidant enzymes are not produced enough when the cell is required? So we looked at the molecular switch level, what is happening to this molecular switch called NRF2, and we found that NRF2 itself is degraded through a variety of mechanisms, through viral infections, and so that if molecular switch that itself is not available, these enzymes are not able to produce enough quantities. So when these are not able to produce in enough quantity, free radicals are not neutralized, and when free radicals are in high amounts, they are going and damaging the cell. And when the epithelial lung cells are not able to function properly, so when in the severe cases of infection, children cannot breathe properly who are infected with RSV. That's how they give oxygen from outside and several other stuff, several treatment. So that's how it is very discomfortable to the children. So now we have identified the mechanism how this oxidative stress takes place in lung cells during RSV infection. So we looked at the various approaches wherein we can reduce the oxidative stress and that might be beneficial to the patients. So we looked at some molecules called NRF2 inducers. NRF2 is a molecular switch. So if you induce the switch to be on and synthesize on antioxidant enzymes, that could be beneficial to the lung cells. So we have used a chemical called BHA. It is a food preservative generally used as, and which also has lot of antioxidant properties. So we treated mice with this chemical called BHA, and in our experimental model, this mice infected with RSV and treated with this chemical BHA showed faster recovery compared to the just infection alone. So we checked at cellular level what is happening. So this chemical is inducing NRF2 production, NRF2 molecular switch, so that it goes and synthesizes increased amounts of antioxidant enzymes, and they neutralize all the free radicals, so that cells will be healthier. When cells are healthier, they recover fast, and they neutralize virus inside. And also we tested dietary supplements. You would have heard a lot about resveratrol, quercetin and all these. So these dietary supplements also have antioxidant properties. So we checked a lung epithelial cells. This is in a cells flask. We tested whether if we treated cells with these dietary supplements, and we infected, and surprisingly we found this antioxidant enzyme synthesis have increased, and cells were healthy. And when cells are healthy, the RSV multiplication is reduced compared to the not treated. So in a nutshell, all these we can say that dietary supplements are good with our antioxidants. When we give to the patients, maybe they recover faster compared to the conventional treatments where there is no specific treatment. So in a nutshell, we have found the mechanism of oxidative stress, and in mice when we treated with BHA, they could recover faster, and even in the lung epithelial cells when we treated with resveratrol, quercetin, oxidative stress could be controlled. So when it comes to the so we have further work. We should work on in human patients how these are going to be helpful. I mean, there may be some obstacles could be there, how this drug to be delivered in the area of lung cells, and how much the kinetics. These are should be worked on by some other groups. And I hope it makes some sense, and you have to understand about this viral infections and oxidative stress. And thank you.